Hello. We're going over my notes for life science and cell biology. In a nutshell. So right now we're going over the what genetic material is. The details. They are DNA strands, deoxyribonucleic acid, which in 3 dimensions is a spiral looking ladder helix molecule. um, it consists of on the vertical chains phosphate, sugar phosphates. On the ladder rungs in the horizontal, you have groups of pyrimidines, either cytosine or thymine, and linking up with the groups of purines, either adenine or guanine. And the way the molecule is shaped in a helix, and you have minor and major grooves, the way it's kinked a little bit because the molecules are not all the same shape, the same size, and shape, whatnot. Summary: DNA is a thread-like chain of nucleotides carrying genetic instructions used in growth, development, functioning, reproduction of all living organisms. Nucleotides are the organic molecules serving as the monomer units for forming the nucleic acid polymers, which is deoxyribonucleic acid. So polymers is a chain of multiple units. The ribonucleic acid, or RNA, is similar, both of which are essential biomolecules in all life forms on Earth. So RNA is ribonucleic acid instead of deoxyribonucleic acid. So I believe, it just has, I think that it gets oxygenated or something. But nucleotides are the building blocks of nucleic acids. They're composed of three subunit molecules: a nitrogenous base, a 5-carbon sugar, at least one phosphate group, okay, so we're just redescribing the molecule. So it contains the sugar, the phosphate group, and the nitrogenous base are called nucleotides. Then RNA and DNA, they're both called nucleic acids. It's alongside proteins, lipids, complex carbohydrates, or polysaccharides. They're one of the four major types of macromolecules that are essential for all known types of life. Most DNA molecules consist of two biopolymer strands coiled around each other to form a double helix. Okay. Each nucleotide is composed of one of 4 nitrogen bases, again, cytosine, guanine, adenine, thymine, and a sugar called deoxyribose in DNA and ribose in RNA, and a phosphate group. The nucleotides are joined by covalent bonds between the sugar of one nucleotide and the phosphate of the next, resulting in alternating sugar-phosphate backbone. The nitrogenous bases of the two separate polynucleotide strands are bound together according to ATCG rule. A and T, and C with G, with hydrogen bonds making double-stranded DNA. And so the hydrogen bonds bind the bases to one another. In a DNA molecule, the pyrimidines, which are thymine and cytosine, bind the purines, which are adenine and guanine. Just the category of molecules. RNA strands are created using DNA strands as a template in a process called transcription. Under the genetic code of DNA, the RNA strands are then translated to specify the sequence of amino acids within proteins in a process called translation. Within eukaryotic cells, DNA is organized into long structures, so the chromosomes. During cell division, the chromosomes are duplicated in DNA replication. Each cell has to have its own complete set of chromosomes. And this is what the replication is for. Eukaryotic organisms, animals, plants, fungi, protists, store most of their DNA inside the nucleus, and some of their DNA in organelles such as mitochondria or chloroplasts. In contrast, prokaryotes, like bacteria and archaea, store their DNA in the cytoplasm. Within the eukaryotic chromosomes, chromatin proteins such as histones compact and organize DNA. These compact structures guide the interaction between DNA and other proteins, helping control which parts of the DNA are transcribed. You have a picture here showing exactly the DNA molecule structure: an adenine molecule, a thymine molecule, cytosine, and a guanine. And you have alternating phosphate and sugar, aka deoxyribose backbone. So phosphate and deoxyribose phosphate. Okay. Here you have showing the way you have I believe is this transcription. So DNA replication occurs when the double helix is unwound by a helicase and topoisomerase. The DNA polymerase molecule produces a leading strand copy of off the chain of the DNA that you're copying. Then another DNA polymerase binds to the lagging strand. This enzyme makes discontinuous segments called Okazaki fragments before DNA ligase joins them together. Okay, so that's the process of transcription. So you have a strand of DNA and structural proteins organize DNA into a compact structure called chromatin. The proteins organize DNA, and in eukaryotes, the structure involves DNA binding to complex of small basic proteins, so the histones, and in prokaryotes, multiple types of proteins are involved in compaction. In orange, you have DNA here, the blue showing the histone protein molecules that make them more condensed. The protein's basic amino acids bind to the acidic phosphate groups. So acidic phosphate groups of DNA are attracted. to